Thanks, Mike.
Okay, there we go. Perfect. Yeah, so what we'll do is we'll start with George then, and it'll do your presentation, and then we'll have time for questions at the end. You have 20 minutes, so I'll start a timer. I'll probably put my hand up. If you can see me, I'll put my hand up, just to let you know when you're getting close to that. So when you get to about 19 minutes. Then open for questions, and we can play it basically however you want. So I think it'd be better if everyone has their videos on when we're doing questions, and then we can ask questions about the paper, and so on and so forth. As I've highlighted before, don't stress too much. You don't know the answer to the question. We're trying to stimulate discussion. We're trying to think about interesting areas. Equally, people might come up with questions that the authors themselves hadn't thought about or don't know. Okay, so let me bring up my paperwork.
Can you see my screen?
Yes, I can see your screen. So let me just bring up the paper so I have that to the side of me as well. Okay, so you can start when you want. Sorry, let me just start a timer. Why can't I not find timer on my phone? All right. Yeah, so if you want to begin, it'll be fine.
Okay, so the paper I've looked at today is the Pyruvate lactate axis modulates cardiac hypertrophy and heart failure, by Clinton et al., published in Cell Metabolism today, so we've got a bit of a sneak preview, I think. So the overview of this paper, it shows that a distinct metabolic event occurs early in cardiac hypertrophy, leading to heart failure, and it introduces a targeted drug to reduce that, to target that metabolic event and reduce hypertrophy, and that drug successfully did reduce hypertrophy in cultured human cells and mice. So the implication for humans of this drug, that the authors really brought up, is that it may help humans respond successfully to left ventricular assist device implantation. And that's mentioned quite a lot throughout this study, that the current standard of care is, even if the heart shows improvement after left ventricular assist device implantation and removal, the standard of care is still a heart transplant. So the improvement is potentially aimed at helping people recover without that heart transplant. And we're going to discuss to what extent that's likely from this therapy, as well as the mechanism that they've found. So the study was on humans, mice, and cultured cells. The human population is heart failure patients with left ventricular assist device before and after the LVAD implantation. So basically this is a very invasive surgery, and it offers researchers an opportunity to collect tissue samples that they wouldn't otherwise have. And they split these humans into responders and non-responders depending on whether their markers improved following LVAD removal. The mice were C57BLAC6, MPC1 conditional knockout mice and control mice, and we'll talk about conditional knockout later if that's not something you're familiar with. The cell cultures were H9C2 human cardiomyocytes and cultured cells from the mice. And these MDA MB231 breast cancer cells were also used for CRISPR screening of the drug. So the human patients were divided by response or non-response to the LVAD. And it was defined by response or not was defined by left ventricular ejection fraction and end diastolic diameter after the removal. So this is a truncated table, and if you could see the whole table, what you would see is there's very little difference, very little way we can tell if someone's going to respond or not until we remove the device, and we see that when they don't respond, the LVEDD is virtually the same as pre-implantation. So here we're looking at, if they do respond, the LVEDD is reduced, and the ejection fraction, moreover, is significantly increased or dramatically increased in responders compared to non-responders. And although it's difficult to have a healthy control because we're obviously taking tissue samples following invasive surgery, they did use donor hearts, so some of our Western blots that we're going to see do have a control group, which is obviously very good. So they showed that the LVAD responders showed increased expression of mitochondrial pyruvate carrier MPC1 and reduced phosphorylation of pyruvate dehydrogenase compared to pre-LVAD. MPC1 is a mitochondrial membrane protein. It transports pyruvate from the cytoplasm to the mitochondrial matrix. So loss of this protein is going to result in a loss of aerobic respiration in the TCA cycle and a comparative increase of lactic acid generating glycolysis or lactate generating glycolysis. And one thing that I found interesting is we have this phosphorylated PDH very high in pre-implantation responders and not so high in pre-implantation non-responders. And that was interesting to me because there doesn't seem to be much of a biomarker to determine if people will respond before you implant the device. And this seems like a possible candidate, but this was not really discussed, and this may be a bit beyond the scope of their study, but certainly possibly interesting to follow up on. What they did discuss was that the responders significantly, as you can see, increased MPC1 expression. These are Western blots quantifications, and the non-responders did not. So that's the protein of interest here. The actual Western blots themselves are not shown in the study, so we're having to take the author's words that these quantifications are correct, which I assume they are. So the next part of the study was to generate these conditional knockout mice, and these mice were littermates, so heterozygous mice were used to generate the CKO mice and the wild-type offspring. Tissue and time-specific knockout of MPC1 was induced by free tamoxifen injections, and then they used RNA-seq to have a look at what happened 8 weeks post-injection and 16 weeks post-injection. And I'm not going to read out all of this, but this is what they describe as metabolic rewiring at 8 weeks, LVEDD dilation at 9 weeks, increase in left ventricular mass at 13 weeks, and fatal heart failure and mitochondrial dysfunction at 16 weeks. And they do include some nice electron microscope pictures of the dysfunctional mitochondria, which I haven't included here, but if you look at the paper, they have that. I wanted to talk a bit about this tamoxifen-inducible conditional knockouts, because it's a method I wasn't particularly familiar with, and I'm not sure if you've come across it before. But what they're doing is Cre is a bacterial recombinase protein, and they're expressing it on a cardiomyocyte-specific promoter, so that's why it's tissue-specific, co-expressing it with a mutant estrogen receptor which inhibits it, and these, when it's induced by tamoxifen and the estrogen receptor is bound to tamoxifen and cannot be bound to Cre, the Cre is then going to bind with this locus X, which again is its bacterial target. It's going to remove part of the gene, a critical part of the gene, excise and recombine it into this circle, which is basically just deleted from the genome. And you can read some more about this here, or there are various other reviews. One of the reasons I wanted to talk about it was because tamoxifen itself is obviously biologically active. It modulates the estrogen receptor activity, and it does have side effects in human patients, and some of these side effects I found were at least tangentially related to the heart, this risk of deep vein thrombosis and blood clots. So they didn't use a tamoxifen negative control, so every mouse in this study was injected by tamoxifen. And I wanted to discuss that. There were only 3 injections. I think this is the justification for not using the control. Only 3 injections were needed to permanently induce the knockout, and obviously all of these mice are going to be sacrificed. The wild type ones also are going to be killed to get the tissue from their hearts. So there's an ethical consideration now with using animals to find a result that may not be necessary. And this study here discusses some of the tamoxifen clearance and injection protocols that make this probably a good method. But still something to be mindful of, especially when tamoxifen might interfere with your actual result. But I do think it's well justified not to use the control here. And they did, of course, inject the wild-type mice with the tamoxifen as well, so they used a control in that respect, importantly. So, MPC1 overexpression. This is in cultured cells now. Reduced hypertrophy from multiple causes. Excuse me, sorry. I did mention that. Yeah, sorry. Yeah, so the overexpression in empty vector control cells were treated with 3 different drugs, plus an MPC1 inhibitor. And hypertrophy was observed in all of these cells, but it was prevented in the MPC1 overexpression cells. Glucose tracing showed increases in intra and extracellular lactate in empty vector cells and decreases in mitochondrial citrate, and that really shows it's a proof of what we assumed before, was that this is shifting the balance towards glycolysis away from aerobic respiration. And also it's important and quite impactful that these 3 drugs act on different signaling pathways that are listed here, and the results were common between all 3 of them. So this is a common event that happens early in cardiac hypertrophy. I think that's a very impactful and important finding. So they generated a monocarboxylate transporter inhibitor, and this is the protein here which exports lactate from the cell. And part of their rationale for doing this was the bidirectional nature of this conversion from pyruvate to lactate, and the fact that alanine was able to enter the mitochondria still at 12 weeks post-injection of the conditional knockout mice. So there is still a way to activate this TCA cycle. So the logic was that if they drastically increased the amount of lactate in the cell, a lot of it would be converted back to pyruvate, and the concentration of pyruvate would result in a reactivation of the TCA cycle, either through pyruvate entering through other methods or through alanine entering and being converted into that. So CRISPR screening of the molecules of the molecule VB124, which is an inhibitor for this monocarboxylate transporter, showed that all the co-lethal genes were involved in oxidative phosphorylation. And moreover, a 180-day trial in mice showed that this drug was safe, at least to use on mice. this is the alanine result we discussed. So the alanine is still able to enter the mitochondria. So I thought this was a really good picture. Again, we're using multiple causes, and these adult mouse cardiomyocytes are clearly, their hypertrophy is being reduced by this drug. In human cultured cells, we get the same response. It not only prevents it, as we see on the left here, but it also reverses it if you allow the phenylephrine to act for 4 days and then add VB124 for 2 days. So there's two potentially impactful findings here. One is the pyruvate lactate axis has been shown to modulate cardiac hypertrophy and heart failure. It seems like cheating because that's the title of the paper, but we will discuss that a bit more. And the novel MCT inhibitor VB124 has been shown to ameliorate cardiac hypertrophy in mice and cultured cells. Again, potentially very impactful. So the impact of establishing a common metabolic event in cardiac hypertrophy and heart failure, I think this is a graphical abstract where they describe their mechanistic finding. I think it's certainly a very good graphic, and it provides a lot of background for other research to expand on what happens at the loss of this MPC. It's a very nice and easy to follow picture. It's also potentially quite incomplete. It doesn't mention what role other mitochondrial carriers play. We certainly mentioned alanine. It doesn't mention other parts of the mechanism, pyruvate to alanine conversion and back in the mitochondria potentially, pyruvate to lactate conversion, and potentially could these parts of the mechanism be targeted by drugs. They've chosen a specific target for the drug, which was successful, or relatively successful, in the limited experiments they did on cultured cells and mice. But if that drug turns out not to be successful, they've still provided a lot of information for people to research in the future, which is obviously really good. So the potential impact of VB124 on heart patients, there's two potential impacts really. If it's an early event in cardiac hypertrophy, could it be administered before heart failure occurs to a large body of patients? Potentially, but in this case we're talking about side effects a lot because you're administering it to a lot of people. It disrupts lactate transport out of the cell. So my first thought on that is, what does it do to the patient's ability to exercise, which is obviously a health intervention that maybe early stage heart disease patients may need. So that's certainly a question. The bigger question that the authors are more concerned with, and they mention a lot, is can VB124 alleviate the need for heart transplant in LVAD patients? This would solve the problem I mentioned because you're giving it to a much smaller number of patients, and one where the intervention can make a very big difference, and they're kind of beyond the point then when exercise is necessarily going to help them reverse their condition. So the side effects and the cost benefit of having side effects may be pushed in favour of taking the drug. Now, as I mentioned before, this current standard of care in post-LVAD patients, even if they're responders, is for heart transplant. And that was the case in this study. So all of their patients, whether they were responders or not, received a heart transplant. They write this hypothetical diagram. It's hypothetical in the sense that they didn't study it, but there are post-LVAD patients who have responded and recovered without heart transplant. And this is a something they intend to focus on in the future. So I've drawn this sort of diagram to demonstrate what I mean, really. If there's three groups of patients, there's non-responders who clearly need a heart transplant because they don't improve. There's responders who improve but need a heart transplant anyway. And then around 15% of people, they say, possibly, because it's not well studied at the moment, it's only a few clinics doing this, can remove the LVAD and recover. So the big sort of £1 million question is, can VB124 move people into this third group? And the point I'm making here is, if it moves people from the non-responder group to the responder group, but they still need a heart transplant, it may have limited impact. If it moves people from the responder group to what I've called the recovering responder group, I think that has a very high impact. It reduces the need for heart transplantation. And they discuss studying this in the future. From the data I've read, I've really gone back and forth on what's more likely, what this is more likely to do. As we saw early in the study, the responders do already have some MCT recovery, MPC1 recovery, excuse me. So that makes me think maybe, it's going to move them from non-responders to responders without alleviating the need for heart transplant. That's possible, but I really don't know, and I think this is a case where more research is needed, and they've stated that that's some of the research they intend to do. If you've read the study, I'd be intrigued to know what you think about that particular question. But otherwise, that's the end of the presentation, and I know we've covered a lot, so I'm happy to go back and discuss anything that you have questions over, clarify anything you have questions over, or start the discussion.
Perfect. Your timing was exact. You hit like 20 minutes to the to the second. Excellent. I think it's a really interesting paper. I think LVAD, as I said, LVAD was never supposed to be a treatment. It was supposed to be that stopgap between you've got heart failure and you're going to die and a transplant. And the idea is that there's more to it. So yes, it could be a treatment, but actually, you've pulled out some nice aspects there that highlight that maybe this drug can make LVAD from simply that stopgap into that more important recovery phase. And I guess the other aspect is, do you think? I guess this is my first question before I open it up. Do you think you could use this drug on people with heart failure without using LVAD? So do you think there's an aspect there that you could try and recover people without having to have the intervention of this, huge metal pump in your chest?
So I sort of brought up, because this is potentially an early stage intervention. I mean, I suppose there's two ways of looking at that question. Either you're using this drug quite early in the heart disease and using it for a long time, or you're using it, quite late and sort of expecting it to be a miracle. Obviously I think, obviously I haven't worded that very charitably, but I think the latter is quite unlikely that it'll work. Although it is possible just from these preliminary researches, but maybe we'll have clinical trials and it'll happen. I think if I go back, so when mice were showing heart failure and fatal heart failure at 16 weeks, I think a lot of these things were irreversible. So the reason I brought up alanine, for example, at 12 weeks is that 12 weeks was really the last point at which that was normal. So I think when heart failure actually occurs, this treatment probably wouldn't rescue people from it. In terms of administering it to people earlier in the heart disease, again, I think it's possible, but that means it's a more long-term dosage, and the cost-benefit of side effects, I think, comes into play more. So, does that answer your question?
Yeah, I think I don't think there's an answer to my question. I think it was more just a thought piece. And the other issue you have here is that these drugs are great, but they're not targeting anything they're not targeting the heart specifically. You can have some isoform differences, but yeah.
That was sort of the point I was making with exercise, because obviously if you inhibit lactate transport out of, for example, skeletal muscle cells, and especially if you're administering it, I was wondering if you have thoughts on that, if you're administering it at a point where exercise might be an important intervention. Is that a side effect to be concerned with? That was just my thoughts. I haven't really read much.
Yeah, I think that's a good question. It's a good point. The difference is going to be at what point in heart failure are you doing it? Because if you're trying to give it to someone towards the end of end stage heart failure, then their ability to exercise is pretty much gone anyway. So you might be able to rescue it. But yeah, you're right. The issue here you have is that these drugs are great, and I'm guilty of it myself. These drugs are great when you're looking specifically at an isolated heart or an isolated cell, but when you're looking in a body, it's different. Anyway, I've taken up lots of questioning time then. Anyone else got anyone's got questions if they want to pop on?
Yeah, I've got one. So how comparable do you think the donor hearts are in comparison? Because obviously they're taking tissue from the LVAD patients.
Yeah. I think the donor hearts are basically as good of a control group as practically you can get, because as I say, you're using this opportunity of invasive surgery to take tissue samples from diseased hearts, and clearly you can't have the control group where you simply give invasive surgery to healthy people and take their tissue. So
yeah, exactly.
I'm trying to think of differences. I mean, because these are tissue samples from hearts, I would say it's a pretty good control group.
Did they have a criteria for the donor heart? So obviously, if it's a control, they're expecting healthy, I'd assume.
Yeah. So these were described as healthy donor hearts, and obviously, since the patients received heart transplants, presumably there's a fairly well-established protocol for making sure the donor hearts are healthy. I don't know exactly what it is, I have to say, but they described these as healthy donor hearts. And,
I assume they might have some kind of background criteria they would have had to, I assume, pass in order to be.
I think it's quite likely that they do.
Yeah.
I'm afraid I can't tell you what it is. Yeah, it does. Does that answer your question? I think they're the
it's quite
donor
interesting.
hearts are about the best control group you could imagine, but I think for our reasons they might not be perfect.
Yeah, it's just interesting to compare.
Yeah.
Yeah, I agree. I think having donor hearts there is quite an interesting way of doing it, because a lot of the time it's a tiny bit of extra tissue that they're doing something else in someone's heart and take a little bit of extra tissue, and so that person isn't healthy because they're having heart surgery. So it's quite a nice one. I think you highlight this figure E It's really curious that
this one here on the
it's
the
really
PDH.
it's really curious those pre-responders have such a high inhibition of PDH, as in
I agree. It's presented in the paper without much comment as well. It really does seem to be beyond the scope of what they wanted to study, is my impression from the fact that their only real comment on this is that PDH inhibition was reduced more in post-responders, and that's obviously true, but they don't mention the fact that it started off just so high.
I think that's exactly. I saw that comment as well. They said, "Oh, yes, PDH activity was really returned very well in the responders, but when you start from such a high point and you go down to a level, I just think it was interesting because you said there's two aspects there. Is there more phosphorylated PDH, or is there more PDH? So because that's normalized to total PDH, maybe there's less PDH there, there's less total, but when you're dividing that by the phosphorylated, it gives you an apparent increase. I don't know. And the answer here would be to use DMP, is one of the things I was talking about in my lectures, because you can measure PDH in vivo in a person, so you could do this.
Can I ask from a layman here, what is the clinical significance for the PDH?
I think it's in this study, it's significant in. Do you mean the sort of clinical significance as in what does it mean for these patients potentially?
Yeah.
Well, I think it's hard to say because they really don't go into much more detail. The reduced PDH inhibition, they expected to be basically they expected the PDH inhibition to be inversely related to this MPC1 expression, because I think more. I'm trying to just get this right in my head, but more pyruvate dehydrogenase is means more pyruvate transport as well, I think. But in terms of clinical significance, I think it's very interesting that there's a significant difference between, especially pre-implant, because as I mentioned, there's really not much. And if you go back to this table, as I mentioned, it was truncated, and there's a lot of other lines to this table where the p-value is like 0.5 or something, so really very little difference in most measurables between responders and non-responders. So the idea that you might be able to give some kind of treatment related to PDH that could make the LVAD more effective, or that you could predict its effectiveness. I suppose, as Mike's mentioned, predicting its effectiveness doesn't have that much value because it's still the stopgap. The patients still need it, otherwise they'll die, and if they're non-responders, they still need it to get their heart transplant. It's not really clear just from this study what the clinical significance is, just because they do kind of gloss over it, to be honest. But I think it potentially has some.
I think you've given me more there, George, than the paper, which is excellent. So thank you. That's really good. That just shows your depth of understanding, which is fantastic. Thank you for that.
Thanks.
Have we got any other questions? Said if you can come on the mic, or
I can ask something.
Yeah, go for it.
The researchers observed that depletion of MPC resulted into an overexpression of gene classes that are involved in cardiac cell development, polygenic organization, and others. Can you bring a possible theory to explain how the loss of MPC can determine the upregulation of those genes?
In very broad strokes, I can. So it's not necessarily the loss of MPC that's directly leading to changes in these gene expressions, but it's the fact that with MPC1 knockout, you're really disrupting the balance of glucose metabolism away from aerobic respiration in the mitochondria and towards anaerobic respiration in the cytoplasm. And I think we've discussed before in our lectures briefly that the metabolites of normal metabolism also function as signaling molecules, so they're going to relate to gene expression. So I think the loss of normal metabolism is leading to changes in gene expression by that mechanism. I can't necessarily list the exact signaling molecules. Acetyl-CoA would probably be one, but I think we've discussed there are others. So I think that's, in broad strokes, the answer to your question. I can't be that much more specific, but I think the signaling cascades that result from normal metabolism here are being disrupted. Does that answer your question, Denisa?
Oh, yes, thank you.
April, I think you had your hand up.
Oh, sorry, I just accidentally shared. I was just wondering, what do you reckon? Why do you reckon they use tamoxifen to do the knockdown rather than like a reversible? Because I know you said that the kind of irreversibility of it like reduced the scope of their controls and stuff. So I just wondered why they wouldn't use like a more reversible knockdown, like whether it's a cost thing or like a methodology reason.
I think my point about the irreversibility was that it actually justified the use of tamoxifen, because if you do something reversible and you need to continually inject a drug to sustain it, so some sort of, for example, you're talking about maybe short hairpin RNA
Yeah, I was thinking
something like that.
RNA interference or something like that.
You
Yeah.
need to constantly inject them to sustain it. If you look at the actual timeline, their use of tamoxifen, it would be here. You have these. I don't know if you can still see my cursor, but you still have you have these 3 injections here, and then the tamoxifen is probably no longer an issue about here, and then all of these events are coming from the loss of the MPC1 gene. Whereas if you were doing something reversible and you were constantly injecting, because they do want permanent knockdown, but the reason they use this conditional knockout method is because if they just use traditional knockout mice, those mice just won't survive. They won't be viable. So they're born with the MPC1 and then they have it knocked down at a certain stage in their life. And then we're measuring the effects of the knockdown. We don't want to measure the effects of the actual injections that they're having. The fact that the last tamoxifen injection is 3 days after the first one, and then you don't have to give them tamoxifen anymore, I think means that you can clearly establish this 8-WPI result as a result of the knockdown and nothing to do with the actual drug they've been given. There are alternatives to tamoxifen. You can use tetracycline, and in that case you would be using it as a sort of positive transcription factor to induce the Cre, and that's an alternative, but I think the use of tamoxifen is quite well justified here. But I brought up the fact that it is biologically active and it does things, just because I think that's something to be mindful of when you're deciding how to generate your conditional knockout mice. But I think their decision to use tamoxifen is well justified here.
Cool, thank you.
Okay, I think unless anyone else has any questions, I think we'll let George rest, have a breath. Nope. No more. Perfect. Well, thank you very much.
Thank you.
And yes, excellent. I'll just stop my recording.